The medical revolution that is embryonic stem cells demonstrate a great potential to cure many diseases. However, due to an extensive debate surrounding the moral status of the embryo, all research has been put to a halt. Imagine, after a long night out, you are driving home and fall asleep at the wheel. Your vehicle going off-road and crashing directly into a tree. You wake up in a white-walled hospital suffering from many serious injuries, one of which is quadriplegia paralyzation. The only hope that you have to use your limbs again is a type of research which involves the killing of the embryo and harvesting its pluripotent cells to repair the damage done to your spinal cord. However, you are denied of this due to a battle of ethics preventing you from receiving the therapy. This nightmare is a reality for many suffering from paralysis. Embryonic stem cells hold a great potential to cure and treat many diseases and injuries, including paralysis, but their research is seen by both the public and some scientists as ethically wrong. So, what are embryonic stem cells, and do their ethical implications hinder the progress of disease treatment? Within our body, there are over 200 different types of cells, each programmed to complete its prescribed job. It is the job of the stem cell to repair and replenish the stemmatic cells when they die. The human body naturally contains stem cells. They can be found, for example, within the bone marrow, liver, and skin. However, the stem cells that are found here are multipotent, which means they can only give rise to a small amount of different cell types. It is the pluripotent nature of the embryonic stem cells which allows them to give rise to any type of cell within the body, and thus they are able to hold a great potential to cure and treat many diseases. To produce embryonic stem cells, fertilization must first occur. This journey of sexual reproduction begins with the sperm entering the vagina and ending its journey in the oviduct through penetrating the female ovum. After this immediate form of prenatal development, the zygote engages in a series of cleavage-like divisions, producing an undifferentiated mass of blastomere cells. In regards to embryonic stem cell research, fertilization occurs through the use of in vitro fertilization, or IVF. A process in which a male sperm cell and a female egg cell are placed together, encouraging fertilization to take place within a culture dish. IVF, a method to treat infertility, produces a various amount of embryos in the case that the first implantation does not create a pregnancy. This results in a large number of excess stored embryos. Statistically, within 2018, there were over 400,000 IVF-produced embryos in frozen storage in the United States. Of this number, approximately 3% will be discarded and will never achieve a full moral status. Utilizing these discarded embryos for research purposes would still lead to the destruction of the embryo. However, their pluripotent nature is extremely valued and would be used by the researcher rather than being completely discarded. If one was to support embryonic stem cell research, they would then also permit the destruction of the embryo. And further, if one opposes embryonic research, one would then be denying any potential benefits of the research. Thus, a problem is created. Credible research that those opposing stem cell research deny could potentially cure diseases such as Alzheimer's, diabetes, spinal cord injuries, and stroke-inflicted damage. And, unlike the drugs that are currently available, stem cells can assist in repairing or replacing damaged tissues, as opposed to delaying symptoms. These cells could also enable us to understand presently unknown diseases. This is done by creating genetically identical embryonic stem cells to the patient and experimenting on these to develop a deeper understanding on the distinct features of that person's disease. This will then essentially create a personalized medicine. Alzheimer's disease is an irreversible brain disorder which gradually degrades and destroys one's memory and cognitive abilities. It is the most common cause of dementia. Its cause, although unknown, is believed to be due to a buildup of amyloid beta protein, which is able to prevent the neurons from sending signals efficiently. Current researchers are utilizing induced pluripotent stem cells to grow neurons with the same genes as those who are affected with Alzheimer's. Using these cells, researchers have been able to model how a patient's body would react to new drugs and treatments that could potentially reduce the amyloid production. Nearing the end of her father's life, Rosie Lamar saw the true effects that Alzheimer's could have on a person, and knowing this, we asked her the question: if utilizing the pluripotent cells within an embryo could cure Alzheimer's, would you take the treatment with the full knowledge that if you did, you would be destroying an embryo?
Yes, I think I would, actually. You're prolonging his life. I think that if it's a guarantee, then I think I would have to try it, and to try and give him that extra time and the dignity of maintaining the person that he was, rather than the frustration that I've seen people go through who's had Alzheimer's, not being able to remember things, and it sort of like belittles them, demeans who they are. That people don't know who they were and what they've done in their life, and they sort of all of a sudden look like they're someone who's just childlike. And I think I would probably a little bit selfishly do it, just so that he could have that that last stage of his life be in a position where he would rather be.
Often, those whom have had to deal with the effects of a disease such as Alzheimer's will choose the side of the problem that utilizes embryos in hope for a cure for their loved one's disease. Using embryonic stem cells to study disease and test drugs rather than using animals or live persons could increase the efficiency of treatment as well as be a safer method of testing, as it would avoid the exposure of patients to experimental drugs. Developing therapies and possible cures for previously incurable diseases is part of the reason as to why embryonic stem cells are such a tremendous recent scientific find. Furthermore, embryonic stem cell research could be key to treating and curing diabetes. Worldwide, 400 million people are living with diabetes, with an expected jump to 600 million by 2035. Diabetes is a condition in which the body cannot regulate or properly use glucose in the blood. In the pancreas, there are hundreds of cluster cells, one of these being beta cells, which produce the hormone insulin. Insulin's job within the body is to signal the other cells to take in the sugar that is within the bloodstream, and therefore it balances the body's sugar levels. For those living with type 1 diabetes, the body does not produce enough insulin. Or, in the case of type 2 diabetes, the cells within the body do not respond to the insulin that is being released and thus result in overly high sugar levels. In the case of diabetes, embryonic stem cells could be used to better understand and assist in creating a cure. For example, using the pluripotent nature of the cell to generate beta cells, which could be used for a beta cell replacement theory. Kylie Buden is a mother of 3 who has had to live with the effects of diabetes every day. Her son, now aged 11, was diagnosed at the age of 2.5 with type 1 diabetes. Knowing this, I asked Kylie the question: if utilizing the pluripotent cells within an embryo would cure diabetes, would you take the treatment with the full knowledge that you would be destroying an embryo in the process?
If the impact was it wasn't to cause someone else pain or suffering, of course. Diabetes is 24/7. It doesn't stop. It doesn't have a rest day. And therefore, if there was a cure, of course I would do whatever it takes to do so.
With such a large impact in her life, it is clear to see why Kylie would take the treatment of embryonic stem cells to cure her son's diabetes. This then provides evidence to denote that those who suffer from a disease such as diabetes will produce a more powerfully swayed view on stem cell research in a hope for a cure, compared to a person who does not suffer from a disease, who are less powerfully swayed. Showing a clear oppositional perspective to the problem is the Roman Catholic Church. Catholicism, or Christianity, is one of the world's most common religions, and it is because of this that their opinion is extremely influential onto the public. Their view on embryonic stem cell research is explicitly stated within the Dignus Persona. The human being is to be respected and treated as a person from the moment of conception, and therefore from that same moment his rights as a person must be recognized, among which in the first place is the inevitable right of every innocent human being to life. The Roman Catholic Church believes that the developing embryo is more than just a collection of cells, but rather a potential human that should have the same moral status and thus the same set of moral rights as an adult human being. This then presents them with a strong moral reason to oppose this research. The perspective provides a paradox that gives the embryo a full moral status and thus saves the life of the embryo, but affects the life of another who suffers from an incurable disease. According to the gene Utaka, it is correct to view embryos in reproductive clinics who are bound either to be discarded or frozen in perpetuity as innocent lives who will die in any case, and those third parties with Alzheimer's, Parkinson's, and other diseases as other innocent lives who may be saved or at least helped by the virtue of research on embryos. Additionally, another perspective believes that it is impermissible to derive and use embryonic stem cells which have been created solely for the purpose of research. This means that the embryo must be donated to scientific research through IVF procedures rather than created purely for the reason of research, only to be destroyed. In the United States, there are approximately 600,000 embryos created for fertility treatments which are now being stored in nitrogen freezers, many of which are not needed for reproduction and therefore have the potential to be presented to scientific research. Further, there are others who hold the perspective that continues to hinder the research of embryonic stem cells. The belief that destroying an unborn child indirectly encourages the murder of other human beings. This statement is clearly flawed in the circumstances of embryonic stem cell research, as the embryos being used are pre-committed to destruction prior to being retrieved by the researcher. Despite this, people are extremely reluctant to change their strong views on the moral status of the embryo. In contrast to these perspectives, a survey out of 50 people that was conducted, 73.8% believe that the potential benefits of the research outweighed the loss of the embryo's life, denoting the concept that because something has the ability to become something else, it does not mean that we should treat it as though it has already reached such potential. Furthermore, the survey presented that although people agree with using embryos for medical research, they also believe that alternative types of stem cell research should be sought after before first considering using embryos. It is this thought of another source which has brought about the alternative to embryonic stem cells. Induced pluripotent cells, or iPS cells, are created by utilizing a stemmatic cell such as a skin cell from an individual and then reverting them through introducing them to a specific gene or molecule, which forces them into a pluripotent state, much like the embryo's own pluripotency. As these cells are not a result of fertilization and cannot result in a human being, they are more accepted by the public. iPS cells have been thoroughly researched over the last 12 years and have shown great promise in developing a pro-life cure to many diseases. PhD candidate Michael Sullivan is researching how iPS cells can assist in creating a cure for Alzheimer's.
My name is Michael. I'm a PhD student in the Cassia Lab at the University of Sydney. In 2007, these two researchers from Japan, Yamanaka and Takahashi, won the Nobel Prize. I think it was awarded in 2012. They won the Nobel Prize for figuring out that you could obtain stem cells without having to source them from an embryonic state. So initially we thought that embryonic differentiation was a one-way process. So you went from an embryonic stem cell to just your adult stem cells, and then they went to whatever terminal cell in the body would use, whether it be a cardiac cell, a skin cell, a brain cell, whatever it is. We thought that was a one-way process. But in 2007, these researchers found out that you could actually introduce just 4 transcription factors, and that turns the cell from your somatic endpoint cell back into a stem cell. And so what these are called is they're called iPSCs. That's short for induced pluripotent stem cells. And so what we use is we use iPSCs to model Alzheimer's, and how we do that is we take the skin cell from an Alzheimer's disease patient, just the regular skin cells, specifically a fibroblast, and we add these 4 transcription factors, and this turns the skin cell back into a stem cell. So at no stage have we really done any harm to any organism throughout this process. We've just taken a little bit of really superfluous tissue from a person's body, and from our stem cell that we get, we can use pretty much the same methods that we would use for an embryonic stem cell. So we turn them into neurons, and we turn them into different cells throughout the body. So that's how our method differs, is we take them from an adult, we reprogram them back into a stem cell stage, and then we use it from there, instead of taking them from the actual embryo itself.
Michael's research presents an ulterior method to treat Alzheimer's without the need for destroying an embryo, and would then therefore adhere and address the different for and against perspectives on the problem. Embryonic stem cells are an unlimited source of pluripotent cells, and therefore are an extremely powerful tool for biomedical research development. However, their unique promise to treat and potentially cure diseases is halted by its ethical considerations. It is because of the considerations surrounding the moral status of the embryo that the problem was created. A problem where one side believes that the potential benefits of the research outweigh the moral rights of the embryo, and the opposing view denies the benefits of the research. Ethical problems have caused researchers to seek ulterior motives to treat diseases in the same way that embryonic stem cells could. Thus, induced pluripotent stem cells were created and now show great potential to cure and treat currently incurable diseases. Despite iPS cells' possibilities, deriving them requires a large amount of time, in contrast to embryonic stem cells, which are more convenient to derive. Therefore, we can conclusively say that due to the ethical implications of embryonic stem cell research, progress on treatment of disease is greatly hindered.